DNA's chemical composition is that of a polymer, made up of many nucleotide monomers. Each nucleotide consists of a nitrogenous base, deoxyribose sugar, and a phosphate group. A nitrogenous base is an organic molecule that contains one or more nitrogen atoms within a heterocyclic ring, giving it basic, alkaline properties. There are two major types determined by the structure of their ring system. Adenine and guanine are purines, formed of a fused bicyclic ring, one 6-membered ring and one 5-membered ring. Both rings contain nitrogen atoms at specific positions which participate in hydrogen bonding. With regard to key functional groups, adenine has an amino group at the 6th position, while guanine has an amino group at the 2 position and a carbonyl group at position 6. Cytosine and thymine, however, are pyrimidines, comprised of a single 6-membered ring, with nitrogen at positions 1 and 3. Regarding functional groups, cytosine has a carbonyl group at C2 and amino group at C4. Thymine has carbonyl groups at both C2 and C4, and a methyl group at C5. It is worth mentioning that uracil is also a pyrimidine, with the same structure as thymine, minus the methyl group at C5. Nitrogenous bases form specific hydrogen bonds in base pairing. Adenine and thymine form 2 hydrogen bonds, while guanine and cytosine form 3, making them the stronger of the pairs. The second component, deoxyribose, is a 5-carbon sugar, which serves as the sugar backbone of DNA. The name deoxy comes from it lacking one oxygen atom compared to ribose, the sugar of RNA. This makes it more stable and less prone to hydrolysis. It is a monosaccharide consisting of a 5-membered ring. The nitrogenous base attaches to the 1 carbon, forming a nucleoside. When phosphate is added to the 5' carbon, it becomes a nucleotide. Lastly is the phosphate group, a polyatomic anion derived from phosphoric acid. Each nucleotide has a phosphate group attached to the 5' carbon of the deoxyribose sugar. This phosphate group connects the 3' carbon of the sugar in the next nucleotide, forming a 3' to 5' phosphodiester bond. A phosphodiester bond is a covalent bond formed between a 3' hydroxyl group of one sugar and the 5' phosphate group of the next sugar. The phosphodiester bond is catalyzed by DNA polymerases during replication.